have Brian Shoichet with us today. Brian Shoichet's as part of the QCRG UCSF AViDD Center and has been trailblazing new methods for computer-aided drug discovery, as well as really discovering some fantastic biology and really interesting confounding factors and experiments for antiviral discovery and other computer-aided drug discovery areas. So without further ado, I'll hand it over to you, Brian.
Okay, terrific. Yeah, it's the latter topic. Well, I don't know how trailblazing it is, but I'm going to talk about the confounding factors in antiviral drug discovery. They're really confounding factors for a lot of aspects of drug discovery, but focus on the antiviral ones. And I'll talk about two common artifacts that we've run into, and I think they're common in the field. The first is colloidal aggregates, colloidal aggregation as a confound in early drug discovery. And I'll talk about its prevalence and how you can control for it. And then I'll talk about how it's impacted antiviral discovery for SARS-2. And in the second part of the talk, if I get to it, talk about a second confound that's particularly prominent for antivirals and I think has had its greatest impact on repurposing, and that's phospholipidosis. We think it actually impacts a lot of areas of drug discovery, but we have most of our data is in antivirals. The colloidal aggregation, the first topic, is something we stumbled into about 20 years ago. It's embarrassing. We were actually working on new compounds discovered by structure-based methods, and they had all sorts of funny properties, which turned out to be very common in the field. We didn't realize it at first, but it turns out that the problems we were struggling with our new structure-based discovered inhibitors for beta-lactamase at the time were seen by everybody. People didn't talk about them publicly so much because they didn't understand really what was going on. Actually, I think a few people actually did. And they had the following properties. You would discover these inhibitors usually from a high throughput screen, and they would, when you controlled for their activities, you found that they were non-specific. When people took the time to dive into it, they found out that they were time-dependent inhibitors, which usually make you think they were covalent, but they weren't covalent. You could show that by rapid reversibility by dilution. They had steep dose response curves. That's something that a lot of the pharmacologists picked up on very quickly. Experiments that we did in our lab showed that they were decreased by increasing enzyme concentration, which is a really weird effect for what's meant to be, things acting in the Michaelis regime, and was hard to explain. And eventually, we struck, because we had run out of hypotheses, we struck on this idea that the molecules might be forming what we called at the time micelles, but people in that field quickly corrected me and said, no, these are colloids. And the thought was that they would form these aggregates, and it was the aggregates that would be responsible for the inhibition of in our case, enzymes, but lots of targets are affected by this. And that was a crazy idea at the time, but it was also appealing because it could be tested directly. And so you could, if these guys were forming sizable particles, you should be able to see them by dynamic light scattering, a technology we'd never really encountered before, but it was, well controlled, very well understood in the field. And sure enough, the particles formed very well behaved spectra in the DLS. They scattered light strongly. In fact, you could, one of my students at the time showed me that you could just do this by eye. You could take a quartz cuvette and shine a laser through it and look at it at 90 degrees. It was important not to look at it dead on, and you would see this blue scattering going on. Well, that's what's going on, and the particles scatter light. And you could also show it by transmission electron microscopy. and the particle sizes were consistent between the two methods, so this became the explanation for these molecules. And we did a lot of other work to characterize them. There's projects still going on. You can show in a fluorescence microscope that, for instance, here's GFP in the fluorescence microscope, sort of under differentiated fields of green because they're all in solution. And then you put, tetraiodo-phenolphthalein in, which is sort of a poster child for colloidal aggregation, and the GFP clumped up, so it formed these punctate features in the fluorescence microscope, and that's because it was being sequestered by the colloids. And one of the techniques we discovered relatively early, a lot of this was just stumbling around, was that non-ionic detergents would in the enzyme assays would disrupt colloid formation. And sure enough, they would do it in the fluorescence microscopy. You had something like Triton X100, which we use very often. There's others that you can use, and both in enzyme, it disrupts both in the enzyme assays and in the microscopy, presumably because it just disrupts the aggregates. So we had, in early papers, we had gotten hold of screening hits from a few drug companies that were behaving weird, and we showed that they also behave weird. But we wanted to do this at scale because the question was, well, is this a little problem in early drug discovery, or is it a big problem in early drug discovery? And the way we tried to do that quantitatively was in collaboration with the NCGC in Rockville, Maryland, led by Jim and Glaze at the time. So what we did is we launched these two large screens, one against beta-lactamase and one against cruzane, with different, the library, what was then the MLSMR, which is effectively the national screening deck, was growing at the time, and we did it with two different instantiations of that deck. And the experiment was the same in both cases. We took the enzyme and screened either 70,000 for beta-lactamase or just under 200,000 for cruzane, and another enzyme, just in buffer for inhibition. And then and when I say it was Jim and his guys at the NCGC did exactly the same experiment with but adding Triton. So exactly all the conditions are the same, but now we add Triton, which is meant to disrupt the colloidal aggregates. And on our side, we did a lot of computation and actually a lot of protein crystallography and structural work and follow-up assays. But anyways, they did most of the heavy lifting. And so the idea was that if these guys were inhibiting, the true inhibitors should not be affected by Triton. That's something we'd shown multiple times, but the colloidal aggregates would be. And then we would just see what percentage of inhibition could be owed to colloidal aggregation. And so this is an example of the data we got. The nice thing about working with the NCGC was that they got beautiful data. They did everything in 7-point dose response, sometimes more, but everything got done. And so it was high throughput screening, but everything in 7-point dose response. Anyway, so this is an example of a well-behaved inhibitor. This is a beta-lactam. As a penicillin, it inhibits beta-lactamase covalently. And you can see that in the presence or absence of Triton, the dose response curve is the same. So that this is not a colloidal aggregator. We don't expect it to be. But this molecule here, which actually is a drug, I just can't remember what drug it is. I think it's actually a beta adrenergic receptor antagonist. But anyways, this molecule in the absence of detergent, which is this red line here, it looks like a pretty good inhibitor of beta-lactamase. But in the presence of detergent, all the inhibition goes away. So that says it's probably an aggregator, and follow-up studies, that turned out to be true. And so overall, so we did this for so beta-lactamase and for cruzane, and this is just like pie charts of the types of inhibitors we found. And for the molecules we found, for beta-lactamase, we actually didn't find any reversible inhibitors whatsoever. They were all, 95% of them were aggregators, some were reproducible. A few were interesting covalents. We got crystal structures of those, and some were beta-lactams, which we knew about. And for cruzane, we actually did find some true ligands, not too many, but in both cases, between 88 and 95% of the hits were colloidal aggregates. So what that teaches is that colloidal aggregation is certainly a major and probably the dominant mechanism of artifact, at least for enzymes and more recently GPCRs in early discovery. So it's a big problem. Okay, so I'm going to transition to what we've done with. I want to set that background. I'll transition to what we found for COVID-19, but I'll take you through the assays we use to check for colloidal aggregation. So, I mean, my pitch is that the problem with colloidal aggregation is it's very widespread, and it's basically impossible to avoid by fixing your library or something like that, because it's just a property of matter, colloid formation. But the good news is that it's really easy to check for it. So it shouldn't, as long as you can control for it, it shouldn't slow you down too much. So we do it multiple ways. We look for particle formation in a DLS. We use a 384 well DLS instrument from Wyatt. You have to tweak it a little bit, but they know how to do that at Wyatt. We run counter screens. So like if we're screening for NSP14 inhibitors, which we are, and we're worried that they might be aggregating, which we always are, we'll counter screen it usually against one or of two enzymes, and sometimes both. A really easy one for the field is malate dehydrogenase. It's got a really easy assay. It's pretty sensitive to colloidal aggregation. You just do it with and without detergent. So this is an example of an aggregator that, in the absence of detergent, inhibits really well. And then you add 0.01% Triton X100 or tween 20, and the inhibition disappears, no longer visible. And this is actually for this aggregator that we found that was published in the literature. I think this is Mpro RdRp. And then, a third but weaker criterion for is it an aggregator, steep dose response curves, steep hill slopes. So this is an example of the same molecule with its dose response curve. And you can see it's pretty steep. You usually want a hill slope of 1. That's not, I mean, there's real reasons you can get higher hill slopes, but they're unusual actually. And that's often in early discovery a sign of a problem. But the problem with the high hill slopes is you can get aggregators that don't have high hill slopes. There's several other things you can do. You can spin, colloids will spin down, proteins won't. So you just put your aggregators in just a benchtop microfuge and spin it down for 20 minutes. It works better if it's in the cold, and then take the supernatant and do that before and after spin down. And if all of your inhibition goes away, it's a good sign you've got colloids. You can increase target concentration. That's what we did early on. It's a little bit clumsy. And by NMR, you can look for peak broadening. And there's in fact other ways of doing it. So there's multiple ways of getting that, like, is my great new hit an aggregator? And the good thing about the DLS and the enzyme assays is they're fast. Okay, so we had a paper about 1.5 years ago on the prevalence of aggregation in early lead discovery for COVID-19. Most of the early papers focused on major protease and the polymerase, and we looked at 56 of the molecules that had been published, and over a third of them were aggregators. They were actually closer to a half of them had problems. There were other problems in them. Some of them were redox cyclers, and there were other funny things going on, but about a third of them were colloidal aggregators. This is one of them, clotrimazole. It's a drug. You think, well, drugs shouldn't aggregate, but drugs are molecules too, and at screen relevant concentrations, low micromolar, they do. This is clotrimazole showing you the malate dehydrogenase counter screen. You see it's got the steep dose dose response curve. It inhibits malate dehydrogenase basically at the same concentrations that it was reported to inhibit, I think, Mpro. And then when you add detergent, this green arrow says, here's what happened, the inhibition drops when you add detergent. And you know, we looked at it by light scatter, and you can, these aren't DLS curves, these are showing critical aggregation concentration, which turns out to be the same range, a little bit lower than the literature value for where it inhibited. And then we did it for, you know, we did it for 19 molecules. I'll show you 4 of them. So manidipine was that molecule I had on the previous slide, same sort of thing, you know, high hill slope. It inhibits malate dehydrogenase actually better than its on-target effect for Mpro. And here's the critical aggregation concentration is about 3 micromole. And the same thing for aripiprazole, also published to inhibit these enzymes. And so these are the first 3 are drugs. They're approved drugs, widely used. Nothing wrong with them at lower concentrations. And this is, you know, a natural product. There's these natural products, often from traditional Chinese medicine or from plant phytols that are very popular for some reason in screening decks, and they're sort of notorious. They have all sorts of problems. I mean, this is a very ugly molecule. It's going to be an oxidation problem, blah, 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 but it's also an aggregator, and that the aggregation is sufficient to explain its activity. It's actually far more potent on malate dehydrogenase than it was on the SARS-2 enzymes. Okay, so that's published. More recently, Izzy Glenn in the lab, a really innovative technician in the lab, has been looking at, these guys all came out of biochemical assays, enzyme inhibition assays, but there's a lot published in cell-based assays looking for inhibiting infectivity, often using pseudovirus. A lot of those molecules look weird to us. Curcumin, if you look up actually, if you look up a lot of these molecules, but look up curcumin, there's like thousands, tens of thousands maybe of papers published on curcumin doing this pharmacologically, or doing that, or doing something else, often in vitro. And it's like, this molecule doesn't do anything. It has no pharmacology. It doesn't get out of the gut. And it's a notorious aggregator, and sure enough, it's in the COVID literature too. So just to show, here's its IC50 on malate dehydrogenase. The red dot says what happens when you add triton. It all goes away, and here it's, you know, strongly scatters. Same thing. This molecule is got a lot of play. It's a derivative of tetrahydrocannabinol of THC. And it was shown to inhibit SARS infectivity, but in the same concentration range that it inhibits SARS infectivity. It inhibits beta-lactamase, which is our AMP-C, which is our second counter-screening enzyme, and it scatters very strongly. And you can actually show this in the flow. So this is similar to the experiment I showed you with GFP. This is spike protein, labeled spike protein with a fluorophore. And it looks, you know, red in the fluorescence microscope, just like undifferentiated field of red. And then you add curcumin to it, and you get these punctate fluorescence consistent with the spike protein being sequestered on the surface of the colloidal aggregate. So this all holds together. Okay, so I think I'm not out of time yet. So I'll try and quickly go through the phospholipidosis side of the story. This is like, these are all stories of like, we stumbled into things very painfully and sunk all our own ships first, and then we started to think, well, are they common? So same story. Nevin Krogan had led this project early in the pandemic to characterize the interactome between SARS proteins and human proteins in the infection cycle. And then we tried, based on what they identified, the human proteins they have identified, we and Kayvon Shoichet and a team of all-stars tried to find molecules that would perturb those. And the two we settled on, unfortunately, are the sigma 1 and sigma 2 proteins, and early on it looked great. We got these molecules. These are well-known molecules for these integral membrane proteins, known to bind to them. What they do is unclear at all. But anyways, and they were active. So it was a great day in the lab. They turned out to be, we knew they inhibited, or they bound to the SARS proteins, one or the other, often both. And sure enough, the red lines are the antiviral activities of these molecules, against COVID-19. So that was very exciting. They were, often sub-micromolar, down to low hundreds of micromolar, and that came out in this paper 3 years ago, basically. And we continue to work on it because there's a huge amount of in vitro pharmacology for the sigma receptors. There are lots of molecules we could get. And after several months of working on it, the painful thing we realized was there was really no SAR molecules that inhibited. So the x-axis here is affinity on sigma 1, and more to the right is more potent, and the y-axis is antiviral EC50 in these cell-based assays. And there was molecules that were super potent, like this guy, Mpro on sigma 1 and sigma 2, actually, and it had no antiviral activity. And then there were these relatively weak ones like chlorpromazine and amiodarone, which weren't that good on the sigma receptors, but super strong in the antiviral assays. And eventually, what I remembered from, all those med chem lectures that I spaced out on was these guys are sort of notorious for inducing phospholipidosis. And phospholipidosis is a disruption of lipid homeostasis. It's a big problem in drug development because it causes late stage tox, and these molecules are well known to do that. So we wondered, okay, is the phospholipidosis behind the antiviral activity? And the characteristic of all of these molecules is they're called cationic amphiphilic drugs. So they've got a cation at physiological pH. They bear a positive charge. They're greasy, and they're known to lead to phospholipidosis. So we went back to the literature and collected from, at the time, looked at some of the high-profile papers, including our own, and collected, 58 molecules from the literature that had been published as, drug repurposing for COVID-19, and depending on how you count, between a third to over 50% of them were these CAD molecules thought to induce phospholipidosis. And we followed that up in experiments with Francois Poignet and Francesca Moretti at Novartis in Basel. And sure enough, what you could show is that in the range where these molecules were antiviral, they would induce phospholipidosis. And this is shown by one at a time fluorescence microscopy. The blue lines are the phospholipidosis, the black lines are cell death. So they don't induce cell death, but they strongly induce phospholipidosis. And you could show that, to a greater or lesser extent, that basically all the hits in the literature were inducing phospholipidosis, all the cationic amphiphiles in these. And then we did it. I'm running out of time, so I won't get through it all. You could show it prospectively too. You could go find new molecules that we hadn't looked at previously, and that actually some of them weren't in the literature as repurposing, but they were cationic amphiphiles, and you could show that they induce phospholipidosis, and they were antiviral. So cationic amphiphile that induces phospholipidosis was reliably antiviral, and the molecules that were not. So I'll just show you this. So there was this good correlation between antiviral. So this is DIPL is drug-induced phospholipidosis on the x-axis, and the y-axis is antiviral activity. And there was a good correlation for all these molecules between antiviral activity and phospholipidosis, whereas the negative controls like Mpro, remember, told you, you probably forgot. I would have forgotten. Melphalan is potent on sigma, but does not induce phospholipidosis. It's not greasy. It's not big and greasy enough, and it is not antiviral. So I was told I had 25 minutes. I'm at 25 minutes. I do have a couple of more slides about new work we're doing. Go for it. Okay, I'll try and finish quickly then. Oh yeah, just to show. Last thing about this project from Tia to me in the lab is that sort of heroic work. Chris White and Adolfo Garcia Sastre at Mount Sinai in New York looked at these CAD molecules to see if they were. I mean, they were inducing phospholipidosis. The phospholipidosis is genuinely antiviral, but not for a good reason. And so they said, well, maybe they'll be active in animals, but they weren't. What they did is they made the animals sick. So like a molecule like remdesivir or emmetine, actually, which also came out of our repurposing studies and others. We're not the only ones. This is actually a pretty potent antiviral. It's an old drug no longer used. And remdesivir is a potent, as everybody knows, a potent antiviral in the mouth. These are mouse studies, but these guys are not significant. What they did is they made the mice sick because we had to load them up to get a good effect. Okay, so this is the one slide. We're going to talk a little bit about mechanism very briefly, but this is the one slide that might be useful to people for the phospholipidosis. And we've developed, actually we didn't develop, we stole an assay that actually the NCGC developed a long time ago. It's a rapid assay for looking for phospholipidosis. It takes 3 days. It's not that rapid. It takes 3 days, but on the day you measure, on day 3, it's a 384 well plate assay. You don't have to do it one by microscopy, and the data are beautiful. That has been published. We've adapted a little bit. We can send anybody the protocol who wants it, but I can also send the paper. And we've been using it to now more broadly test these CADs, not only. So one of the things we want to say is, well, it's not just COVID-19. There's a huge literature on drug repurposing for antivirals. This is Ebola data, but we've done the part of that's Chris White has done a lot of others, and they're effective basically against every virus we've looked at. I wanted to say one quick thing about mechanism. So one thing that's emerged is sort of this cholesterol axis. Because we're into what's going on? Why does phospholipidosis is a huge problem for pharma, but its mechanism isn't really well understood. And so we thought we'd look at that, and we'd like to, if we understand the mechanism, we can control for it better pragmatically. And so one thing that Alex White, who was a postdoc in the lab, noticed is that phenotypically Nieman-Pick disease has a lot of the same phenomena that we see in phospholipidosis. So he noticed 2 things. One is that there's a cryostructure of the Nieman-Pick protein, NPC1, bounds to this drug itraconazole. And itraconazole is not a CAD. It's not cationic at physiology. It's greasy enough, but it's not cationic. Nevertheless, it as predicted, does induce phospholipidosis, and then when you knock, so this is amiodarone on the right here. When you knock out MPC1, amiodarone, which in the wild type cells, it's the poster child as far as everybody's concerned for phospholipidosis. It does not induce phospholipidosis, and this other MPC1 ligand, which is a cationic amphiphile, and we strong phospholipidosis inducer. When you knock out MPC1, the effect goes away. That's consistent with MPC1 being involved. The MPC1 is a cholesterol transporter. It transports cholesterol out of the lysosome, where the phospholipidosis is thought to, that's the sort of the center of phospholipidosis in the cell. And you can also do it by mevastatin. So if you add mevastatin in lipoprotein depleted media, you can you blunt, you basically, so the black line is the drug, in this case sertraline, inducing phospholipidosis in just normal media, and the blue line is add 50 nanomolar mevastatin, this cholesterol biosynthesis inhibitor, and this is the same thing for this U18 molecule, and here's amiodarone, the poster child. We did it for lots of different molecules. Okay, so I've used up all my time. I'm sorry, but just to remind you that both colloidal aggregation and phospholipidosis are really widespread in antiviral drug discovery. That's the bad news. The good news is certainly for colloidal aggregation, we understand the mechanism very well, and there's easy controls that you can use to rapidly get rid of them or control for it. And phospholipidosis is less well understood, but I think we're making some progress there too. And there too, the assays are straightforward, and we're hopeful. What we hope is we're developing tool molecules that allow the field to perturb it rapidly and decide without, having to go crazy to figure out what's going on. Okay, I'm done.